This call is now being recorded.
Nahid madam, can we start?
Hello. Yes. Yes. You can start.
Aslam sir is joining, madam.
Just I'll find. I'll find.
Yeah, okay, okay.
Hello everyone. We will soon start the session. We are just waiting for our president. As soon as he joins, we will start immediately.
ये अपना शेयर नहीं कर रहे।
So we are starting the session soon. Dear participants, thank you for your patience. And cooperation.
आपके पास What's your color? Okay. We are visible also.
I'm audible. Should I start,
Yes, madam.
madam?
Yes, yes, madam. You are audible.
Nahid madam is there. We will start now.
Nahid, madam.
Nahid madam, shall we start? Good afternoon, sir. Kothari sir.
Good afternoon to you.
Good afternoon. How are you?
Yeah.
This
Well, you are
will-
waiting for some more people to join, or your president
Sir,
to
we are
join?
just waiting for our principal madam to join and our president, sir.
Oh, right. Okay. Not a problem. Rest of the things are all set.
Yes, yes, sir.
Oh, great.
Everything is fine.
And it's all online, or your students are sitting in the hall also?
No, no, sir. It is all online.
Online. Okay.
Nobody is there.
Good, good. Yeah. And you are Archana.
Yes, yes, sir. I am Archana.
Good to see you. Yeah, Archana.
It's nice to meet you, sir. Today.
And is my audio video okay?
Yes, sir. It is good.
And sound quality is okay?
Yes, it is very fine. Good, good.
ठीक है.
Nahid, madam joined.
Yes, yes, sir. Good evening, everyone. I'm there. I'm Dr. Nahid Azam. Hello, sir. How are you? Hello, Kothari,
Hello,
sir.
madam. Good to see
Hello.
you.
Thank you, sir. Very nice to see you, sir.
Thanks a lot. Yeah.
We are delaying the. Ashna madam will start.
Should I start? Okay. Okay.
Yes, ma'am.
Aslam sir.
Sir will be joining after some time.
Yes. Good afternoon, sir. Aslam sir has also joined.
Okay, sir. Okay.
Sir, okay, we will start now. Good afternoon and a very warm welcome to one and all present here. On behalf of KMS Society, GM Women's College, and on behalf of Department of Botany, I, Dr. Archana Gupte from Department of Botany, feel honored and privileged to welcome resource person, principal, president of KMS Society, management members, participants from all over India, my colleagues and my dear students for the national webinar on genetic modification technology and its impact in 21st century. Some insight and future prospects, organized by the Botany Department of Konkan Muslim Education Society, GM Women's College. Before we start, as per our tradition, we take the blessings of Almighty. So I request Ms. Arfa Narvil, student of SYBSc, to recite the Kirat. Over to Arfa.
Assalamu alaikum and a very good afternoon to one and all present here. A'udhu billahi minash shaitanir rajim. Bismillahir rahmanir rahim. Ar-Rahman 'allama al-Qur'an. خلق الإنسان علمه البيان الشمس والقمر بحسبان والنجم والشجر يسجدان والسماء رفعها ووضع الميزان ألا تطغوا في الميزان وأقيموا الوزن بالقسط ولا تخسروا الميزان والأرض وضعها للأنام فيها فاكهة والنخل ذات الأكمام والحب ذو العصف والريحان فبأي آلاء ربكما تكذبان صدق الله العلي العظيم. Over to you, Archana ma'am.
Thank you, Arfa, for making the atmosphere calm and blessed. Now I request all the participants of the webinar to stand at the place they are for national anthem, which will be recited by Ms. Arfa Narvil, a student of SYPS. Ms. Arfa Narvil.
जन गण मन अधिनायक जय हे भारत भाग्य विधाता पंजाब सिंध गुजरात मराठा द्राविड उत्कल बंगा विंध्य हिमाचल यमुना गंगा उच्छल जलधि तरंगा तव शुभ नामे जागे तव शुभ आशीष मागे गाहे तव जय गाथा जन गण मंगल दायक जय हे भारत भाग्य विधाता जय हे जय हे जय हे जय 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 हे. Thank you.
Thank you, Arfa. Thank you all. KMS Society GM Women's College is fortunate to have a kind-hearted, supportive, and encouraging principal, Dr. Nahid Azam. हयात लेके चलो, कायनात लेके चलो, हयात लेके चलो, कायनात लेके चलो, चलो तो सारे जमाने को साथ लेके चलो. Our principal is the person who always believes in teamwork. I request Dr. Nahid Azam, Principal of GM Women's College, to welcome the gathering. Over to you, Dr. Nahid Madam.
Nahid ma'am, unmute yourself, ma'am. Nahid ma'am.
Okay. Yeah. I'm sorry. Thank you very much. Thank you very much, Archana madam. A very good evening to all. Firstly, I would like to congratulate and wish all the best to our organizing committee for the national webinar on genetic modification, technology and its impact in 21st century, some insight and future prospect. A beautiful topic they have chosen. We have incredibly talented, hardworking, talent team and a very supportive management. We are growing under the guidance of Mr. Aslam Faki, our president. He is always there to help us and guide us wherever we are stuck. Before the program starts, I just want to say thanks to our team, Mrs. Lakshmi Shichengla, head of the botany department, Dr. Archana Gupta, Dr. Ritu Jain, Dr. Vaishali Nirmalkar, and star coordinator, Mrs. Jayashri Thakre, all the non-teaching staff of botany department, and a long list of dedicated team from Office of GM Women's College. On behalf of KME Society GM Women's College, I take opportunity to welcome all the participants from India and abroad. I extend a warm welcome to our today's renowned scientist, our resource person, Dr. S.L. Kothari from University of Rajasthan, Jaipur. He had contributed so much in the field of science, especially botany. You will hear shortly about his biodata. I'm not going to read. It's a lengthy one, sir. I hope all of you will be very delighted to hear from him and gather enough information which will help you to grow in the field of research, academic. You may also ask question participants. Everyone, please take this opportunity to ask question from him. He will be the right person today to answer many of your doubts. Really, sir, we are fortunate to have you here. Thank you very much for sparing your time. God bless. Stay safe. Thank you very much. Thank you, Archana madam, for giving me opportunity to speak. Thank you.
Thank you, madam, for your encouraging words. The fundamental pillar of KMS Society, President Mr. Aslam Faki, whose consistent efforts with great vision has achieved number of milestones one by one for the institute. This is the result of his flexibility of mind, the groundedness of purpose, the openness to new ideas, and his dynamic leadership. I would like to dedicate a share for him. जिस दिन से चला हूं मेरी मंजिल पे नजर है, जिस दिन से चला हूं मेरी मंजिल पे नजर है, आंखों ने कभी मील का पत्थर नहीं देखा. May I request the President of KMS Society, Mr. Aslam Faki, to address the participants. Mr. Aslam, sir. Sir, please unmute yourself. Aslam. Aslam sir, please unmute yourself, sir.
Hello.
Yes. Now,
Hello. Can
now,
you hear me now?
yes, sir. Now it
Yes.
is fine. Yes.
I really welcome all of you, and my blessings will always be with you all. And when I heard about this seminar, I was really very happy that your genetic modification technology, a subject which is very important in this time. I am really thankful to SL Kothare for giving his valuable time from Rajasthan. I hope sometimes he will be able to visit our campus and
I
see
will.
for himself. I wish all of you a great success for this seminar. Thank you very much. Thanks a lot. Hello.
Yes. Thank you, sir. Thank you, sir, for your motivational talk. Ours is a world with growing interconnections and interdependencies that reach across all borders. Advances in science and technology continues to improve human lives. Incidentally, the topic for today's webinar is also concerned with the same. We have an eminent personality, Dr. S.L. Kuthari, as our resource person. I request Mrs. Lakshmi Chengala, Head of the Botany Department, to introduce today's speaker. Over to Lakshmi Madam.
Thank you, Archana Madam. Good afternoon, one and all present here. Welcome to this national webinar. I am honored and privileged to introduce to you all today's speaker for the webinar, Professor S.L. Khotare, sir. Sir is a distinguished professor and vice president of ASTIF Amity University, Rajasthan, Jaipur. Sir's specialization is in plant sciences, biotechnology, nanobiotechnology, molecular biology, and plant tissue culture. Sir has completed his PhD from University of Rajasthan in the year 1984. Before joining Amity University, Sir has held various positions at the University of Rajasthan, Jaipur. Sir was Dean of Faculty of Science, Director of Center for Converging Technologies, Director for PG School of Life Sciences, Head Department of Botany, Syndicate member. Sir has been coordinator for DST Pursue program, DBT BIF program, DBT IPLS program, DST FIST program, UGC SAP, UGC UPE program. Sir has received many laurels and held various positions abroad. Sir has been fellow of Fulbright Scholar, University of Illinois, USA, Commonwealth Academic Staff Fellow, University of Nottingham, UK, Rockefeller Biotech Career Fellow, University of Nottingham, UK. Sir has been visiting Foreign Research Fellow at University of Tsukuba, Japan. He had been adjunct professor at RMIT University, Melbourne, Australia. INS visiting fellow at IPK, Germany. Sir is also a fellow of National Academy of Science, National Academy of Agriculture Science, Indian Botanical Society, International Society of Plant Morphologists. Sir has guided 52 students for PhD degree. Sir has 190 research papers to his credit. Total 4,600 citations. He has 40 chapters in books. Sir has completed 18 research projects. Sir has also received Nahar Samman Award from Rajasthani Welfare Association, Mumbai in the year 1997. Sir, we are very fortunate and blessed to have you as our resource person. Thank you for accepting our invitation, sir. Archana Madam, please take over.
Thank you.
Thank you, Mrs. Lakshmi, for introducing Sir. We are fortunate to have Dr. S.L. Kothari with us, whose in-depth knowledge in the field of genetic modification technology will definitely benefit you all. I would like to read a शेर for him. है मेरी परवास के तेवर नए, है मेरी परवास के तेवर नए,
और नाम नहीं मेरा
है मेरे परवास के तेवर नए, साथ के मंजर नए. So it is with special excitement that I am pleased to welcome Dr. S.L. Kothari to conduct the technical session. Over to you, Dr. Kothari, sir.
Thank you very much, Dr. Gupte, for inviting me to this webinar. At the very outset, I would like to thank KME Society and GM Momin Women's College for giving me this opportunity to interact with the faculty and the students of the Women's College, Mumbai, on a topic which I have been pursuing for last more than 25 years. So I'll tell you some depth about what genetic modification technology is and what is its impact in the 21st century and what are the future prospects of this technology. I'm thankful to Aslam Paki, who has very kindly graced this occasion and very politely and very humbly wished this webinar well. Thank you so much, sir. And I also want to thank Dr. Musadik Patel, who is the Honorary Secretary of the Society. Then my thanks and special regards goes to Dr. Nahid Azam, with whom I was in correspondence for this webinar. And I'm sorry that I had to change the date of this webinar, but Madam has been very gracious to assign another date for this event. Dr. Chengala Lakshmi Shri has very vividly and very kindly introduced me to the audience. I'm grateful to you, Madam, HOD Botany Department. Archana Gupte is conducting this session very well and in a very nice, systematic manner. I am grateful to Dr. Ritu Jain because of her insistence that this event is taking place here, and Dr. Vaishali Nirmalkar, who is co-convener of this event, and other faculty members of Momin College, Mumbai, and various faculty members, students, and other people who are joining this webinar from many places. I would like to share the seminar PPT with you now, and in an hour's time, I will complete my talk, and then we will have time for interaction and for any question-answer session. Students and faculty who want to interact with me can write their questions, and later on, we will discuss them all. The topic, as you can see on the screen, genetic modification technology and its impact in the 21st century, some insight and future prospects. Genetic modification technology is not limited to the plants. It is also there for animals, microbes, bacteria, fungi, and viruses. It follows the same principles for plants also as it follows for the animals and microbes. Since I am a plant biologist, my focus will be on genetic transformation and genetic modification of the crop plants. The GM technology has already impacted the society in the 21st century. Lots of products which are there in the market, they come through genetic engineering or recombinant DNA technology. Recombinant DNA technology is also nothing but GM technology. And the products, I can name few examples, such as human insulin, which comes from the genetically engineered E. coli or the yeast, and that is a recombinant DNA product. Hepatitis B vaccine, blood clotting factor 8, golden rice, BT cotton, herbicide resistant various crop plants. All these are examples of GM technology. which is impacting the 21st century in a very, very big manner. To add to this year's Nobel Prize has been given to two ladies who discovered genome editing technology using CRISPR-Cas, Emmanuel Charpentier from Berlin and Jennifer Doudna from University of California at Berkeley. They have discovered CRISPR-Cas technology, and this technology is going to change the face of the earth in the next 10-20 years. Various kinds of genetic modifications and various kinds of genetic diseases in humans and improvement of crop plants will see unprecedented growth in the near future. The genetic modification technology, if I say, then it is a recombinant DNA technology. And what is recombinant DNA technology? It is defined here. Recombinant DNA technology is the joining together of DNA molecules from different organisms and inserting it into a host organism to produce something new genetic combination that are of value to science, medicine, agriculture, and industry. Since it is a technology, therefore it has to be valuable to any commercial product, and these commercial products come from medicine, agriculture, and industry. It can be important for scientific advancement also. That's why science has also been written here. So in a nutshell, the recombinant DNA technology uses DNA from one organism into another organism to produce something useful there. This is an amalgamation of several techniques, recombinant DNA technology. And of these several techniques, it became possible because of the step number 1, isolation of genetic material. And this isolation of genetic material became possible because of the discovery of endonucleases. And these endonucleases, as you know, science students and biology people, it can cut the DNA into smaller fragments at a particular site, and then cutting the gene at the recognition sites, cloning of DNA into a vector, or amplifying the gene copies through polymerase chain reaction. You first isolate the genetic material, then you have to cut it with the restriction endonuclease, then you have to clone it in a vector or through PCR, then join together the DNA fragments through the involvement of the enzyme ligase, and then insert that recombinant DNA into a host cell, and that host cell then should make recombinant products. And the 6th step is to isolate and separate the recombinant cells from the cells which are non-transformed. These are essential 6 steps followed for genetic engineering or genetic modification of either plant or bacteria or virus, or for that matter, animals. So these essential steps are the same for all the organisms. Having said that, I would like then to tell you about genetic modification technology per se. Before that, there are some fundamental questions which all biology people, biology students, and biology teachers should know and appreciate. These fundamental questions which I am going to address in this lecture are: how and why could GM technology become possible? How GM technology is playing God. You are transforming the living organisms on earth, which nobody else can do. Only God has created the life on earth, and we are changing it. So some people feel that we are playing God, but we are not. I will explain to you later. And there is variety of life forms, and these variety of life forms are important for genetic engineering technology. Then what is life and how its continuity is maintained? This continuity of life is also very important to understand why GM technology became possible. And is there any commonality there? Is there any commonality in varied forms of life present on this earth? These fundamental questions I will quickly explain to you, and then I will come to the GM technology. per se. Variety of life forms. You know how many different species and plants and animals and bacteria are present on Earth. So far, we have been able to decipher and catalog only 10 to 15% of the organisms present on Earth and only less than 10% living species present in the ocean. It is estimated that there are about 1.2 crore species of living organisms present on Earth and in the ocean. Of these, only 12 lakh species on Earth and 2 lakh species in the ocean have been catalogued. So of the 1.2 crore, we know only about 10%. That makes to about 12 lakh, or to say it exactly, 14 lakh. And scientists have estimated the projected number of species which we are going to identify in the due course of time. So, in a nutshell, there are 1.2 crore species on Earth, but out of these, only 14 lakh have been identified. About 90% have been identified. About 10% have been identified. And all these life forms have a commonality. And that commonality tells you that these life forms have come from something common. Either common ancestor, or the life forms have come from some chemicals which later on became life, and then that life perpetuated. To give you some figures about the variety of life forms, here is a table which has recently in the 2011 PLOS Biology published this table about the species on Earth and ocean. On the right side there is ocean, and in the middle the Earth, the eukaryotes, animalia, species of animals, 9,53,000, but predicted species is 77 lakhs. Chromista, there are 13,000 cataloged, but 27,000 might be there on the Earth. And in the ocean, this number is 4,000 and 7,000 predicted in the ocean. Fungi, the figure you can see, plenty, protozoa, and total of 12 lakh species on Earth have been catalogued, but 87 lakh species have yet to be discovered and nomenclature. In the ocean, 1,93,000 species have been catalogued, but 22 lakh species are yet to be discovered. Amongst the prokaryote, archaea, the ancient bacteria, 502 species are known, and 455 more have been predicted. Bacterial species, you can see the number, and total numbers and grand total numbers. If you look at the grand total number, the total catalogued species comes out to be 12 plus 2, 14 lakhs, while the predicted and catalogued put together, that makes it about 1.3 crores species. You might wonder what is Chromista. Chromista are algae, but they have chlorophyll C, and they do not store food as starch material. Giant sea kelps, diatoms, they come under this category, and some other fungi also come under the Chromista category, which are colorless. So I told you about variety of life forms, and so this is the number of variety of life forms, and some photograph you can see here how many different types of plants, flowering plants, and vegetables, pteridophytes, bryophytes, animals, single-celled animals, multicellular animals, colonial forms of animals, birds, reptiles, mammals, all sorts of varieties of life forms are present on the earth and in the ocean. And these, looking at them, one wonders how they might have evolved. But if you look deep into them, then you find that there is a commonality in them. And that commonality is very important. And we want to know what is life and how its continuity is maintained. And is there any commonality there? So the commonality is that all life forms are based on cells. Cellular cells are the basic units of every life. And if you can change a cell, then you can change the whole organism. Cells are the basic structural and functional units of life. And the continuity of life occurs through single cell. An organism may be multicellular or very complex organism like elephant or tiger or human being themselves, but all of them start from a single cell. And their life continues in the form of single cell. Cells arise from pre-existing cells. Therefore, if there is no cell, there cannot be any daughter cell. But first cell must have originated by accident or by the circumstances which are there on earth at that time, and this must have come from chemicals which are completely non-living. Genetic basis of life also resides in the cell. So the commonality among all life forms is that they are all based on cells. Why I am telling all these things in a lecture on genetic modification? Because we are going to modify a single cell and then make the whole organism, and we are going to take advantage of all sorts of varieties of life to take their genetic material and make use of it in changing the cells which we want, we desire. All right? And the cellular basis and cells, cellular basis is the basis for every life, but cells are of different types to perform different functions required by an organism. For example, just few examples, cells that connect body parts or cover and line organs, epithelial cells. They are present on the outer covering of your skin, outer covering of all your organs, kidney, liver, everything. The outer covering is made of epithelial cell. Fibroblasts are the cells which make the connective tissue and collagens. Erythrocytes are nucleus-less cells, but they are carriers of hemoglobin and they carry oxygen with them. Then there are cells that move organs and body parts, skeletal muscles and smooth muscle cells. They make the skeletal muscles make the muscle fiber, and smooth muscle cells provide more flexibility to the muscular cells. Then cells that store nutrients, fat cells, are present in the tissue, adipose tissue, and these are adipocytes which store the fat material for future use as food. Macrophages are types of WBCs, and they engulf the apoptotic cells or any pathogenic organism that may have entered your body. Then there are nerve cells, which are very huge cells, very big in size, in length. They have a body and the axon and dendrites. At the bottom, there is a sperm cell, which is a haploid cell of any diploid organism, which is a head and tail, and the function of this sperm cell is to reproduce. So, variety of functions make variety of cells. In the plants also, there are variety of cells, parenchyma, which are living cells, and thin-walled cells. Collenchyma are also living, but they are thick-walled cells, and they do not have much of the intercellular spaces. There are fiber cells, sclerenchyma cells, there are sclerids, there are tracheids, vessels, phloem cells, and above all, there are apical meristematic cells, which are responsible for the growth and development of the plants into whole organism. Cellular basis of the life and different types of cells in living organisms, plants and animals. That is the commonality in all living organisms. All living organisms are based on cells, and cells have common organs in all the plants and all the animals. Plants have a cell wall, while animals do not have a wall. The rest of the things plants and animals also share. Why a biology student might wonder why there is so much of commonality, why every cell is similar, why every cell should have a nucleus, why should mitochondria, plastid, chloroplast, and endoplasmic reticulum, ribosomes, they are all present in all living cells. That must strike you that there is a commonality. And this commonality is the cause for genetic modification becoming possible at the hands of the human. And how then the cell perpetuates and how the essence of life is maintained. Essence of life is maintained through chromosomes, which are present in the nucleus of every cell. The chromosomes, the metaphysic chromosomes are very big, and they can go to the size of up to 700 to 1000 nanometers. Then there are supercoiled structures lower to them. Then there are tight helicular fibril structures. Then nucleosomes are present in the chromosome. Then there are linker DNAs to join the nucleosomes, chromatids, and then comes the DNA, which is about 2 nanometers in diameter. And that DNA is the essence of life. It is a non-living chemical, but it makes the life possible and continuity of life possible and continuity of the continuity of the phenotypic characters possible in all living organisms, while it itself is a non-living thing. DNA is nothing, deoxyribonucleic acid, it is double-stranded, it has deoxyribose sugar, then it has nitrogenous bases, adenine, thymine, cytosine, and guanine. These are all chemicals. And the DNA is the only chemical in the world, living and non-living, which has self-duplicating properties. It can replicate and make its true copy. And this is the reason why life became possible on Earth. This is a chemical, the constitution of which is written here. And this is the only chemical, this and RNA also you can say, but RNA also changes into cDNA and then it duplicates and then it translates, transcribes and translates. So this chemical become duplicate through replication and two strands are formed which each duplicates itself. And that is why the life sustains itself, life maintains its purity and its characteristic features generation after generation. And this continuity of life is maintained by DNA, but at the cellular level, this is done by the division of the cells. Cells divide by a process called mitosis, and through mitosis, the same daughter cells are formed, which are genetically identical to the dividing cells, to the mother cell. And they are identical, and that is why the purity of race is maintained and purity of the cells is maintained, in spite of they being similar, but they perform many different kinds of functions as I have shown you earlier. So they differentiate differently because of the expression of genes at different times in a differential manner. How and why could GM technology become possible? By now, you must have realized what is life, how it is maintained, how continuity of life is maintained through division of the cells. DNA is responsible for maintaining the characteristic features. GM technology became possible at the hands of the mankind because the essential genetic material in any organism is the same. So if it is the same DNA in man and DNA in monkey or DNA in a pig or DNA in a buffalo is the same, or in the bacteria is the same, virus is the same. So man thought that can we transfer the bacterial DNA into the man or the plant and it work? It should work, and it works.
Hello.
That is, Yeah.
Kindly share slides again. Kindly share slides again.
Okay, sir. Can you see them now?
Not visible.
Can you
Thank
see
sir.
my slides now?
Not yet, sir.
When did it stop showing?
No, sir. Actually, someone has shared the screen. Someone has
Yes.
sent earlier. That is why it happened.
शेयर क्यों नहीं हो रहा? सभी को नहीं दिख रही है, आप ही को नहीं दिख रही?
Someone has shared the screen.
अच्छा.
Someone has shared the screen, sir.
अच्छा, अच्छा, अच्छा. Oh.
That is.
For all, not it is not visible, sir. Now visible, sir.
तो शुरू से ही नहीं दिख रहे? किसी ने बीच में कर दिया, स्क्रीन शेयर अपना. क्यों कर दिया?
No, it was visible since the beginning. Just last 1 minute it was.
अच्छा, okay. 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 Right. Is it okay now?
Yes, sir. Yes, sir. It is okay. It is fine.
Great.
I mean, last 1
आप
minute.
Yes, sir, it is visible now.
All right. Great. I'm glad. तो आप मुझे रोक भी सकते हैं अगर कहीं पे कोई दिक्कत हो, तो ये नहीं कि आप सुनते रहें, मैं बोलता रहूं और आपको दिखे फिर भी नहीं. So how and why could GM technology become possible? I was telling about this thing, and it became possible because the genetic material is same in all the living organisms. Plus, nature has done it before we did it. Nothing is possible on this earth which nature may not have done it before us. So nature has done it through what is called as horizontal gene transfer. And this horizontal gene transfer is responsible for the creation of web of life, which we see in a variety of life forms. Horizontal gene transfer is the sharing of genetic material between organisms that are not in a parent-offspring relationship. Horizontal gene transfer means the gene is transferred between two organisms which are sexually not compatible. HGT, horizontal gene transfer, is an adaptive mechanism in bacteria and archaea. It happens between prokaryotes and eukaryotes, prokaryotes and prokaryotes, eukaryotes and eukaryotes, and prokaryotes and eukaryotes. It happens in nature, but it happens more so among more related species. Since it is happening in nature, we also could replicate it. And this horizontal gene transfer takes place through conjugation. You can see on the left side, through conjugation, one bacterial individual is transferring single-stranded DNA into another bacteria, which is receiving it. Two cells can fuse, and through fusion, both the cells can exchange their genetic material with each other. Conjugation, transduction, you know that the genetic material is transferred from one bacteria to another through the agency of virus. And these viruses act as gene transfer agents, and specific DNA also they can transfer into the bacteria. Transformation is also possible in nature. The naked DNA, which may be present in nature, is taken up by bacteria and it gets transformed. Then intracellular or endosymbiotic gene transfer is also possible. In nature, some eukaryotic cells have engulfed prokaryotic cells and they have become resident in the eukaryotic cells. And the horizontal gene transfer is also possible through what is called as introgression. On the left side, you see population A, which is reproducing and maintaining itself. Population B is also maintaining itself. But human intervention, through human intervention, or in nature also, some cross-fertilization can take place, and some of the genetic material from A can go into the B. Right? So because of the HGT, genetic modification became possible. This HGT, many horizontal gene transfer events are neutral, but can adapt to become beneficial to the recipient. the horizontal gene transfer takes place in nature. Most of these horizontal gene transferred into recipient cell are useless, and recipient cell will either discard it or will die. But some of the genes transferred through this mechanism may be useful, and they are retained by the recipient genome, and it is perpetuated. HGT events involve mobile genetic elements present in bacteria and all other organisms that promote their own dissemination by associating with adaptive traits in the gene pool of the mobilome. Mobile genetic elements, all the mobile genetic elements present in a living organism, in a species or in a haploid genome is called as mobilome. HGT is most frequent between closely related species, I told you. Large multicellular eukaryotes have evolved through gene acquisition. Multicellular living organisms, eukaryotes, they have acquired large number of genes through this method of HGT. In the long past, endosymbiotic gene transfer, there are evidence available that bacteria have been engulfed by the living cells and they have become mitochondria or chloroplasts. Novel traits evolve through horizontal gene transfer, prompt adaptive radiation to exploit the new resource without competition. And this is an evolutionary advantage in having HGT. Now, after this brief introduction of genetic transformation technology, I will quickly pass through the genetic transformation technology per se. Kya hai genetic transformation technology? Before that, GM technology, particularly crop genetic transformation, has impacted 21st century in a great manner. First genetically transformed or GM crop was grown in the world in 1996, and since then GM technology has gone to several million hectares of land, and these are the figures for 1917, but 1918 figure have also come, and this figure 189.8 million hectares has become 192 approximately million hectares of land has been cultivated with GM seeds, and this million hectares of land with GM seeds have been grown for herbicide tolerance and for insect tolerance, and combined herbicide and insect tolerance both. And these are some of the crops: cotton, soybean, maize, and canola. Most of the cotton, 80% of the cotton grown in the world is biotech. Soybean 77% and maize 32%, canola 30%. So adoption of GM by farmers is increasing year after year because of its benefits. Global area of biotech crops in million hectares, maize, soybean, cotton, and canola has been shown here. Just to tell you the impact of GM and GM plants which are available to farmers or through research, the GM plants have been produced in alfalfa, apple, canola, beans, carnation, chicory, cotton, cowpea, maize grass, eggplant, eucalyptus, flax, maize, melon, papaya, petunia, pineapple, plum, polished canola, poplar, potato, rice, rose, safflower, soybean, squash, sugar beet, sugar cane, sweet pepper, tobacco, tomato, and wheat. This is the list of GM plants which have been created, which have been developed through what is called as GM technology. What kind of features have been used for commercial cultivation of the GM crops? Mostly it is abiotic stress tolerance, altered growth or altered yield, for disease resistance, for herbicide resistance, for insect resistance, and modified product quality and pollination control in some cases. And what countries have approved GM events? Many countries have approved GM, partially or fully. Argentina, Australia, Bangladesh, Bolivia, Brazil, Burkina Faso, Canada, Chile, China, Colombia, Costa Rica, Cuba, Egypt, Ethiopia, European Union, Honduras, India. Not everything can be grown in India, but cotton has been approved. Indonesia, Iran, Japan, Malaysia, Mexico, Myanmar, New Zealand, Nigeria, Norway, and others. All these countries are listed here. They have approved GM events for growing in their country. Now I come to genetic transformation technology. Genetic transformation technology, in the beginning I have said that we have to follow certain steps. So for genetic transformation technology, if you have to understand it, we have to know what are transgenic plants, why do you want to make them, how to make, the regulatory process, what are the risks, future transgenic crops. So what are transgenic plants? A transgenic plant or genetically modified plant contains one or more genes which have been artificially inserted instead of the plant acquiring them through pollination. The inserted gene sequence may come from another unrelated plant or from a completely different species. Plants containing transgenes are also called as genetically modified or genetically engineered plants. This is the definition of transgenic plants. Nothing, but they contain one or more gene from another species. Is transgenic technology an extension of traditional plant breeding, or is it a revolutionary new development? So transgenic plant they have one or more gene from another species. So transgenic technology is different from plant breeding. Yes, it is different from plant breeding. But it is also, some of the principles of plant breeding are followed by transgenic technology also. Transgenic technology is different in the sense that it draws upon genetic variation across kingdoms rather than within a species or genus. In the plant breeding, we mix genetic material within the same species. But in the transgenic, you can take the genetic material from any source. Gene transfer is more precise in transgenic technology. But both transgenic technology and plant breeding, they depend on the variations. And both these technologies make use of variations. While the plant breeding, hybridization, can draw upon a range of germplasm resources, which is primary and secondary, but transgenic technology can take genetic material from tertiary gene pool also. Here I have shown a slide to tell you about 3 different types of gene pools: primary, secondary, and tertiary. Primary gene pool comes from all the land races of a species, and through sexual hybridization and meiotic recombination, some change in the genetic material is achieved. While the secondary gene pool comes from all the wild species of a genera. For example, rice, Oryza sativa. The primary gene pool will come from Oryza sativa only. But the secondary gene pool will come from Oryza nivara, Oryza rufipogon, Oryza akingeri, Oryza glabrima. There are 20 species of Oryza, and out of which 19 are wild species. Any genetic material which comes into Oryza sativa from the wild species is a secondary gene pool. And the secondary gene pool also requires meiotic recombination, sexual hybridization, but it will be done through human intervention or somatic hybridization through fusing the protoplast of two different species of rice are fused together and secondary gene pool can be brought together into the same cell. In the tertiary gene pool, any DNA sequence present in any source, like thousands of species are there. I told you that there are 12 lakh species which have been catalogued, 14 lakh species. All the genetic material present in all the 14 lakh species comes under tertiary gene pool. This gene pool was of no use for crop improvement because you cannot transfer the genetic material from bacteria into the plant. But through this genetic transformation technology, tertiary gene pool can also be put into the plant or animal of interest. And that is the revolution this technology has brought about 40 years back. And since then, we have been adding numbers to the species which can be genetically modified or transformed. So, to summarize, transgenic crops can contain genetic constructs from any source. This technology allows movement of genes across a species barrier. This is the key thing. Sometimes people ask, do transgenic plants look different from normal plants? No, they look just the same, except with some additional gene or gene function. There is some phenotypic difference at the chemical level in the transgenic plant. But if you want to modify it completely and make it different, looking different from the original species, it is also possible. These are transgenic tomato looking the same as normal tomato, but they have a gene introduced into them so that there will be delay in ripening and shelf life of these tomato has increased. Why do we want to make transgenic plants? In an earlier slide I have shown it. You want to improve the abiotic or biotic stress tolerance in plants, or you want to improve the quality of the product. For that reason, or you want to increase the yield, you make transgenic plants. And transgenic plants have become more important because in the 21st century, the agriculture is facing lots of challenges. What are those? The 21st century has the population of the world grown to something like 7.4 billion people. And to feed those billion people, 7.4 billion people, crop yields must be increased to meet the increasing demand. Use of resources such as land, labor, capital, energy, water, and agrochemical must be reduced. If you reduce these, then the productivity goes down. But you have to reduce them because you don't have extra land, labor is very expensive, money is not available to the farmer, energy is very costly, it causes environmental problems, water is scarce, and agrochemicals interfere with the soil health. And you have to grow crops in drought, heat, cold, and other stresses. These are other challenges. The use of pesticide, herbicide, fertilizer must be reduced to save the environment. All these factors work counterproductive in agriculture because if you do not use pesticide, there is problem. But you have to reduce it. To address all these challenges, genetic transformation technology gives some answer. And that is why this technology has become very popular. Although GM technology has faced so many oppositions ever since it came into existence in 1996. Many countries without any knowledge are opposing GM. And many people opposing GM without ever knowing what biology is. You see on the left side, the BT brinjal on the right side, while the normal brinjal on the left. only one gene has been introduced into the right side, that is BTKM1 variety, with CRY1AC gene introduced into them, and the brinjal are heavy. The insect cannot attack them, and they are more desirable by the consumer. But at the same time, botanists and people also argue that we have so many different kinds of brinjal varieties, round, white, green, yellow, all sorts of brinjals are there. Therefore, if we resort to this GM technology, then everybody will grow these brinjals only because of the consumer preference. And many of the brinjals, which otherwise have aesthetic value and nutritional value and also the taste value, they will not, nobody will grow. That is also right, but that responsibility should lie with the government or with the other people, not with the farmer. If farmer gets benefit by growing brinjal on the right side, he should be allowed to grow. While preserving all other varieties of brinjal should be the responsibility of National Bureau of Plant Genetic Resources or somebody. These are golden rice. They look just the same as normal rice, but they have three extra genes, phytoin synthase, phytoin desaturase, and carotene desaturase. These three genes are not present in the rice plant. For that reason, rice cannot convert isopentenyl pyrophosphate into carotenoids. It can convert IPP into inorganic pyrophosphate, which requires the intervention of phytoin synthase to convert into phytoin, and which is then converted into carotenoids. This technology has been developed, and these three genes coming from bacteria and other sources, daffodils, have been introduced into rice, and rice plant can now produce carotenes, which provides vitamin A, which is a precursor for vitamin A. Transgenic soybean on the right side, you can see. On the left side, there are so many weedy plants growing with soybean. If you spray herbicide, then all weeds will die and the plant will survive better. And the dying weeds will leave nutrition and humidity for the growing crop plant. BT cotton, you can see, is with one gene, and cotton is happy. How to make transgenic? कैसे transgenic plant बनाएं? Students often want to know what is the method for making transgenic plants. So first thing you have to do is isolate and clone the gene of interest which you want to introduce. Say, for example, CRY1AC gene or CRY gene. Add DNA segment to initiate or enhance gene expression. Then add selectable marker to select the cells which have been transformed. Then introduce gene construct into plant cell, select transformed plant cells or tissues, and regenerate whole plant. These are different steps which are required for making transgenic plants. Why are we able to make transgenic plant? Because essence of life, as I told you, is the same in all the living organisms. It is the DNA, which through transcription make RNA, and through translation make protein, and protein expresses in the form of a trait. So DNA, which is present anywhere, can be put into the plant of interest, and that DNA will express into the plant, and its chemotype will be improved. There are four ways in which the genetic material can be introduced into the plant cell: through agrobacterium, through biologistic or microprojectile gene gun. We can also use protoplast, and there is another technology which is recently developed called CRISPR-Cas technology, for which this year's Nobel Prize has been given in chemistry to two scientists from Germany and America, Emmanuel Charpentier from Berlin and Jennifer Doudna from UC Berkeley. They have got the Nobel Prize for their work on CRISPR-Cas system. Agrobacterium tumefaciens, students know that how agrobacterium can transfer its tDNA into the plant cell. What it does is, are my slides visible?
Yes, sir. Yes, sir. They are visible.
Okay. it's becoming a monologue, but I can't help it. After this monologue, we will have dialogue.
Yes, sir.
Plant cell gets wounded, and acetylsyringones are produced by the plant cells, and these acetylsyringones go into the bacteria and they stimulate the Vir genes, which produce single-stranded tDNA, and this tDNA goes into the plant cell. And this, after going into the plant cell, it gets into the nucleus of the plant cell and integrates with the resident genome of the plant cell. And there, the tDNA expresses itself by producing opines, cytokinins, and auxins. Because of the production of auxin and cytokinin, the plant cell divides unlimited and a gall is produced. The bacteria, agrobacterium, stays in the gall and it feeds on the opines produced by the gall cells, galls. In this is a natural genetic engineer. Horizontal gene transfer was done by bacteria into the plant. From the prokaryote to the eukaryote, nature shifts DNA. Scientists thought, can we cut the tDNA and introduce the gene of interest into it, and can the same procedure bacteria will follow, and it follows. And that became the hallmark for the agrobacterium-mediated genetic transformation of the plant cells. Another method is biologistic, or what is called a gene gun. On the left, you see the photograph of a gene gun. In this, at a very high pressure, very small nano or microparticles of tungsten or gold, which are coated with the naked DNA, are shot into the intact plant tissues, maybe a shoot bud or a seed or any callus cells. And at a very high velocity, the gold or platinum particles coated with DNA goes into the cell and they release their DNA into the cell. And that DNA gets incorporated with the recipient cell and that DNA expresses. Now, if you have transformed the plant cell, then you have to find out whether that plant cell has been transformed through southern blot hybridization, where you can see the presence of the foreign DNA. Then northern blot will tell you whether the foreign DNA is making messenger RNA or not. And the western blot will tell whether the protein is being produced or not. And the whole plants then can be regenerated. And there is third method. So sorry, this slide should come afterwards. Third method of genetic transformation is through protoplast. Protoplasts are plant cells which are deprived of their cell wall and they become naked. Because cells are not having cell wall, so they can take up the naked DNA by a chemical agency or through an electrical current. On the right side, you see the rice protoplast. Rice protoplasts are capable of dividing and making full plants. The transformed rice protoplast on the right side, they divide and they can now be cultured. And on the right side, you see all the colonies produced from transformed protoplast making small colonies. Left side is non-transformed, so everything is died. But on the right side, few colonies are coming. They are genetically modified colonies. And they can be regenerated into full plants, which this is rice plant which has got a gene introduced through protoplast technique and polyethylene glycol chemical treatment. The rice plants which have been transformed look much the same as the normal plant, and they also set seeds, have flowers, they have fertile pollen grains, and they look just the same, except one feature which is extra present in them is the tolerance for antibiotic. And that gene, presence of that gene can be verified through what is called a southern hybridization technique. On the left side, first lane you see, that is the marker lane, and the second is a negative control, third lane is a positive control, and then 1, 2, 3, 4, 5, these are transformed plant which have been cut with the restriction enzyme, with 2 enzymes, and that way you see that in the control there is nothing present, while in the transformed plants genetic material is visible. Here is a control experiment done for Illucine coragana. On the bottom right, you see everything is growing. These are callus tissues put on a medium which they like to grow. They are growing. On the top right, the petri dish had the similar pieces of callus, but it is all died because of the presence of some antibiotic. On the left side, top and bottom, both these plates have also been cultured with the same callus tissue, but they have been treated with agrobacterium, which has got NPT2 resistance gene. Some of the colonies are coming up. These are genetically modified colonies. Now these colonies can be separated and can be grown into complete plants. These are genetically modified finger millet plants. And the presence of the gene, which is beta-glucuronidase, Gus gene, can be seen through histochemical analysis. Fourth method of genetic transformation is through genome editing, that is called CRISPR-Cas technology, which has been explained here. But I would refer you to read this paper, the Nobel Prize in Chemistry 2020, Popular Science Background, Genetic Scissors, a tool for rewriting the code of life. Emmanuel Charpentier and Jennifer Doudna have won the Nobel Prize in Chemistry this year for their discovery of CRISPR-Cas technology. All of you should read this to understand this technology, because this is the technology of the future genetic engineering of bacteria, plants, animals, including human beings. This technology, CRISPR-Cas technology, can cause mutations of some utility. They can edit the gene, correct the gene sequence. They can create complex animal models. The technology can be used for knowing the function of the gene. Technology will also allow you to turn on and off specific genes. Genetic modification of the plants is also possible through CRISPR-Cas technology, and in future you may not require protoplast or particle gun or agrobacterium. CRISPR-Cas alone will do. Modification of human germline is also possible, and some of the genetic defects in humans can be corrected at the zygote stage or at the embryonal stage. Benefits of GM technology is an old slide, but it has allowed the reduction of chemical pesticide by 37%. It has increased profit by 22%, farmer profit by 68% in some cases, and increased crop productivity to the tune of $133.3 billion since the adoption of this technology. So thank you very much. I hope I have been able to explain to you the genetic modification technology and its impact in 21st century in an introduction manner. Now the talk will be open for any questions. Thank you very much.
Thank you, sir, for your enlightening presentation. I hope it will surely useful to the researcher in this field. Now the forum is open for participants. They have already posted questions in the chat box. I request Dr. Vaishali Nirmalkar to conduct the interaction session. Dr. Vaishali.
Yes, thank you, ma'am. Very inspiring and enlightening talk, sir. Thank you very much for delivering such an important aspect for our students and for our participants. Now there are few questions, sir. So with your kind permission, may I proceed with the question and answer session?
Yeah,
Sorry,
please.
sir.
Yes, please.
Thank you, sir. So first question is, Dr. Leela Chauhan is asking, HGT and somatic hybridization, is it interspecific or intraspecific?
Please say it again.
HGT and somatic hybridization, is it interspecific or intraspecific?
Somatic hybridization is possible, interspecific also, intraspecific also. If it is between two different species, then it will create horizontal gene transfer. Protoplast from the same species can be fused together. I did an experiment in which I fused the protoplast from carrot, two different cell lines of carrot, and fused them together. This was intraspecific hybridization. At the same time, tomato and potato have also been fused together to produce what is called as tomato, and that is interspecific hybridization.
Yes. Thank you, sir. Sir, we have the next question from Dr. Mukesh Pimpliskar. He's asking why it is common belief in scientific circle that research needs to be continued to assess the risk and benefit of crop modified.
Research must be continued.
Yes. I'll again read, sir. Why it is common belief in scientific circle that research needs to be continued to assess the risk and benefit of crops modified. Means, sir, the meaning is that whatever genetically modified crops are there, why there is always a continuous need that we need to
every time you have to use another modification. Because it is the nature, rule of nature that if you are producing, say, insect-resistant cotton and you have grown it for 3-4 years, insect will also develop resistance. And then you have to introduce another gene and another variety will have to be produced. That is true for any other crop, for any even hybridization also. There is a development of resistance in the pathogens also. That is why you will require new variety after some time. Did I answer the question?
Yes, sir. Yes, sir. Sir, the next question is asked by Dr. Nandita Singh. In spite of so much research going on in GM technology, many places and sectors, there are still doubts about long-term safety effect to environment. So, can you give some highlights on that, sir?
This is a very common question and very relevant and very good question. The thing is, before the genetically modified crops are released for farmers' field, all safety considerations are scientifically done, and when it is correctly assessed, thereafter only GM plants are allowed to be grown in the farmers' field. So there is no problem. If the safety measures are properly adhered to, then there is no problem to the environment or long-term effects of the genetically modified plants. If you are eating the carrot gene into rice, it is not going to make any difference.
Yes, correct, sir. Absolutely right. Sir, we have the next question asked by Dr. Dhruvi Nirmal. What is the status of GM crops in India? What is the response? Yeah, sir, she has asked 3 questions. What is the status of GM crops in India? The next question is, what is the response of farmers and policymakers in approving the use of other GM plants apart from BT cotton? And what are your insights on this?
Yeah, who asked this question?
Dr. Dhruvi Nirmal.
Dr. Dhruvi, it's a very good question. The current status of GM technology in India, GM cotton is grown, GM brinjal was ready for cultivation, but it has not received approval. Other than that, any crop plant, rice, wheat, maize, barley, soybean, any plant you name, and Indian scientists can genetically modify it if required. But in the last few years, the interest has gone down because the regulatory aspects are so unfriendly that people lost some interest in GM production. But the BT policy, her second question was about farmers' response.
Yes,
Yeah, farmers are buying GM through even smuggled routes. I heard that lots of farmers in Punjab are growing BT brinjal, and the BT brinjal is not allowed in India, but they are smuggling seeds from Bangladesh. So farmers are much interested in GM, but the anti-GM lobby does not allow this to happen. And BT farmers have definitely benefited from growing BT cotton, tremendously benefited. That is why they are growing. If you ask farmer to buy expensive seed, they will not buy until and unless they are benefited. But environmentalists and other people are fighting against farmers. They don't allow farmers to benefit from the GM. They are asking some questions which are biologically irrelevant.
correct.
Yeah.
Absolutely right, sir. Sir, we have a couple of questions more. Can I proceed with it, sir?
Yeah, most welcome. No problem.
Yeah.
I have no issue.
Dr. Rudresh has asked, please tell me about the applications of hybridoma technology and soma clonal variations in crop improvement.
Yes, soma clonal variations and hybridoma, तो आप जानते ही हैं, hybridoma technology have been utilized for production of monoclonal antibodies, and hybridoma technology is there for so many years. Soma clonal variations are the variations produced in tissue cultures. Okay, so any variation, what plant breeding needs? Variation, good variations. So if soma clonal variations are good variations, then they can be used. And in sugarcane, some good soma clonal variations have been identified, and they have been used for improving the sugarcane.
Yeah.
Yeah.
And sir, the last question is, Dr. Nandita Singh has asked, the CRISPR-Cas technology, can it cause some genetic instability in future?
No. It can create genetic modification. It can create, it can remove some bad genes. It can add some good genes. This is what it can do. But if you want to misuse it, misuse is also possible.
So one more question has appeared here. Dr. Prasanna Kumar Badde has asked. Dr. Prasanna Kumar Badde has asked, do you agree that the plants created using novel genome editing methods like CRISPR technology must be classed as genetically modified organism?
It's a wonderful question. Genetically modified organisms have their genetic material modified. So through CRISPR-Cas also you modify the genetic material. In that sense, it is genetically modified. But if you are taking the genetic material of rice and changing it, then the genetic material is not coming from another species. In that respect, it is not genetically modified. You can say it both ways. But genetically modified, when you do hybridization, sexual hybridization in plant breeding, there also you are making some changes. Some combinations are being changed. Some recombinations are taking place. So modification is taking place there also. So strictly speaking, that is also genetically modified. But here, genetic modification ka matlab hai when you are changing the genetic material through the recombinant DNA technology. Genetic material is taken from one source and put into another source. So CRISPR-Cas, it is not transgenic but cisgenic. Same genome ko change kare, that is cisgenesis, not a transgenesis. But CRISPR-Cas technology can also bring genes from another source and do it, then it is transgenesis.
Okay. Sir, the another question asked by Smita Talwar. Is there any training for GM crops for farmers?
Farmer ko GM crops technology, farmer will never be able to do it. So there is no need of any technology, but farmers are educated about GM technology. What are the benefits, how it works, what it will do. All these things are there. That is why farmers are growing BT cotton. Farmers are much more smart than us. They will not accept anything which is not good for them. But on behalf of farmers, others are raising slogans that it's not good for farmer, while farmer is not telling it. If farmer tells, then farmer had not to tell anybody, he will not use it.
Yes, sir. Yes. And sir, we'll take the last question now. It is asked again by Smita Talwar. Are there any government rules and regulations regarding GM crops?
Oh, certainly. Genetic engineering approval committee is there, and it has to go through several stages before the genetically engineered modified crops go into the farmer's field. So there is most stringent regulations are there in India for GM.
Yes. Thank you very much, sir. Over to you, Archana ma'am.
Thank you, sir, for solving all the doubts of the participants. I think all are clear about genetic modification technology now. Thank you, Vaishali. We are proud to say that our department is selected for Star DBT scheme of Government of India. May I request
Congratulations.
Thank you, sir. May I request Mrs. Jayashree Thakre, the coordinator of Star DBT scheme, to offer concluding remark. Over to Mrs. Jayashree Thakre, madam.
Thank you, Archana madam. Respected President Sir Shri Aslam Faki sir, our dear President, Principal Dr. Nahid Azam madam, and our excellent resource person Dr. Kothari sir, and my dear participants and students. Good evening all, and hope you are doing well. Sir, our college has been selected for the prestigious Star College scheme of DBT, Ministry of Science and Technology. Currently, 5 departments are there: chemistry, botany, zoology, physics, and IT. The scheme is focusing
Congratulations to you and your president, Faki sir.
Yes, sir. Yes, sir.
Because
He is
of his
always
involvement,
yes, sir. He is always guiding us in all our endeavors. And our scheme is focusing on strengthening the undergraduate science teaching. We have been motivating our students to take up challenges in life, and this session will definitely help us in that also.
Our request participant to please mute.
Our India, sir, maximum area is under rural area where agriculture is main occupation. As we are facing pandemic situation, virus is causing the disease, and like that also many plant pathogens cause lot of diseases, and maximum loss is there for farmers. Hence, genetically modified crop is the most important one of the solution for it. However, future safety for consumption should be known to us, and hence the botany department has organized the program to create the awareness, which is very really good initiative, and our students will be benefited through this program. I congratulate the botany department and thank you resource person Dr. S.L. Kothari for enlightening the important topic, which is a great need to improve our agricultural technology, which is beneficial to our country and the nation at large. I once again thank our management members and the principal for giving me this opportunity as a star coordinator, and hope in the future also we will take the benefit to create this awareness and benefit for the community. And thank you very much once again. Stay safe, stay well. Thank you.
Thank you, Jayashree madam, for your good remarks. A success of any event is always a result of well-organized collaborated work. May I request Dr. Ritu Jain to propose formal vote of thanks?
A very good afternoon to one and all present here. On behalf of management, principal, and my own behalf, I am here to propose formal vote of thanks. First of all, I would like to thank our president, Mr. Aslam Fakir sir, a very outstanding personality who created excellent academic environment from KG to PhD level in KME society. Sir, thank you very much. I would like to express my profound gratitude to in charge principal, Dr. Nahid Azam Madam, for the motivation and support. I would like to thank resource person of today's webinar, Dr. S.L. Kothari. Human so meticulous and humble. I insisted him to visit our college someday when I met him. And today in this pandemic, we have got an opportunity to meet you virtually. Thank you, sir. My sincere thanks to DBT for funding. My hearty thanks to STAR coordinator, Mrs. Jayashree Thakre Madam. I would also like to thank our HOD, Mrs. Lakshmi Shri Madam, for her wholehearted involvement and maintaining the environment of whole webinar. A very creative and artistic person by nature. I am happy to express thanks to Dr. Archana Gupte and Dr. Vaishali Nirmalkar. We are also grateful to advisory committee. I would like to thank student volunteers, Ms. Hafsa, Ms. Arfa, and Ms. Malika. My sincere gratitude to all my colleagues who directly or indirectly contributed for making it a grand success. At last, I am thankful to all participants. We have participation from all over the country and abroad from Texas and Dubai. Thank you and have a nice day. Thank you. Over to you, Archana Madam.
Thank you, Madam. I think it's a very enlightening and very nice presentation sir has given to us. I thank you, Aslam sir. I thank you, Nahid Madam. I thanks Dr. S.L. Kothari sir for giving this very good presentation and enlightening the students and enlightening us also, the research students and I think the teachers also has got very much information from the presentation you have given. Thank you all for this webinar. Thanks a lot.
Thank you, madam. Thanks a lot.
There is an announcement
thank you
for
so
the
much.
participants. I request all the participants to fill the feedback form correctly with the correct spelling and kindly wait for the certificates. You will get the certificates within 10 days.
Madam.
Thank you.
Yes, sir.
Madam, please send the feedback form. I have missed it.
Sir, we will be posting in the WhatsApp group also.
Okay.
Yeah, we will be doing that. So don't worry, you will get your certificates also. Thank you, Kothari sir, so much for accepting our invitation, sir.
Thank you, madam.
सर हम लोग बहुत चाह रहे थे कि आप हमारे कॉलेज को विजिट करें सर
I will visit sometime.
जब ये पैंडेमिक खत्म हो जाएगा सर हम जरूर चाहेंगे कि आप हमारे कॉलेज विजिट कीजिए.
I visit our MIT University, so I will visit your college certainly.
Yes, sir. Thank you so much, sir. Thank you so much, sir.
All the best. All the best.
Yeah, thank you so much. Yeah, thank you so much, sir.
I request all the participants along with Kothari sir, for the photograph. All the participants, please switch on the video for the photograph.
Ritu madam, can you please tell Kothari sir to please join again? For the photograph?
Madam.
Yes, madam. Yes, madam.
Lakshmi madam, Naik madam also she has left.
Yeah. I'll inform.
Aslam sir, he is also not there.
The feedback link is open. Participants kindly fill the feedback form.
Kothari sir is
Hello,
joining.
madam.
Okay, okay.
Hello, madam.
Jayashree
Hello.
madam is also left. Mahit madam, Jayashree madam, Lakshmi madam, all three are not there.
Hello. Hello, madam.
Hello.
In the feedback forum, there is no option for filling the college name, madam.
College name option not. It is there.
Hey,
It is there.
only it shows option 2. That's it. But
Vaishali?
when click on when
I'll
click on
recheck
option
it.
2, it is not opening for mention the college name.
Just a minute, sir. I'll recheck it and I'll again post it.
Please check it, madam.
Yeah.
Yes, madam. College name is not there.
Okay, I'll recheck it, sir. Don't worry. I'll again post it.
Vaishali madam, you can start taking the photographs.
Vaishali, open your video also.
Just a minute. I'm just checking the feedback link.
See, others are there.
Done or more photographs are required? Done?
Yes.
Sir, the option for the feedback link is there. For the name of the institute, it is there, sir.
Option 2 is written, madam. Option 2.
Yeah, option 2. So in the option 2, you have to type, sir.
But typing option is not showing, madam.
Thank you.
I have filled the feedback form. Only select option is there. I have submitted also.
Lakshmi.
Okay, just a minute, sir. I'll just change it. Thank you for informing.
Vaishali, photograph.
Just a minute, ma'am.
Yeah, I have taken few. Yeah,
Okay.
I have taken few.
Thank you.
Yes, I have done it. Yeah.
Sir, the feedback form I'm resending. Kindly check this. Those who faced problem, please check it.
Thank you.
Yes. Yeah. Thank you all. I have taken that. Vaishali madam, are you done with the photo? Yeah.
Yes, sir. Yes, ma'am. Yes, ma'am. I hope now the participants
Yes.
don't have any problem in filling the feedback form.
Yeah.
ये बंद नहीं होगा अपना शायद.
Madam, later on you will change that certificate name.
Yes.
Because now it is showing already it is responded. Now it is not showing the form to open.
Okay.
Ma'am, it is showing you have already filled this form.
Okay, ma'am, if you have any other email ID, you can again fill it. The same option goes for all. If you have any other email ID, you can fill the form again. Okay. In any cases, all the participants will get the certificates. Don't worry. If you have any problems, you kindly email us. No issue. Thank you all. Thank you very much.
Okay, madam.
Thank you, sir. Over, no? Vaishali madam, anything else?
No, ma'am.
Okay, Lakshmi madam.
आपका म्यूट है मैडम, लक्ष्मी मैडम आपका म्यूट है और आप बात कर रहे हो.
ओके सॉरी. या एक और प्रिंट में एक और ले रही हूँ मैं स्क्रीनशॉट. ओके या
All participants, no issues. If you have any problem while filling the feedback form, no issues. We will clarify all the things and we will issue the certificate. Don't worry.
On WhatsApp group and on email, we will clarify it. Okay.
yes, photographs are done, madam. Photographs are done. Yeah. Yes, ma'am. Thank you all. Thank you so much, all the participants, for making this program a success. Thank you so much. Any problem, we are there. You can just pin us. We'll get back to you. No worries.
I already get the certificate, ma'am.
Your name has come properly, sir? Your college
Yes,
name
madam.
your college name has also come properly?
Please, madam, 1 second. I check. 1 second.
Yeah. Yeah. Please check.
Yes, madam. All information is correct.
Okay. Thank you so much. Thank you so much.
Yes, ma'am. I too have got this certificate. It's fine. It's perfectly fine.
Thank you. Thank you so much, sir. Thank you.
Good evening, madam. Me also got the certificate with the name and institute name. Thank you very much,
Okay.
ma'am.
Thank you. Thank you.
And this is
Yeah.
for the first time I'm seeing that after filling the feedback, instantly we are getting this certificate. Thank you for this.
You're welcome, sir. You're welcome. You're
Yeah.
welcome. You're
Thank you so
welcome,
much.
sir. Yeah, yeah. Thanks. Thanks for joining. Thanks for joining us. Thank you so much.
Thanks, madam.
Yeah, yeah. Thank you. Thank you.
Hello, madam.
Yes.
Madam, in upcoming days, please conduct the webinars on plant taxonomy, plant pathology related aspects, madam.
You want us to conduct, sir?
No, madam.
You are
you
conducting?
conduct.
अच्छा, you want us to? आप चाहते हैं कि हम कंडक्ट करें या आप करने वाले हैं?
Please tell me in English, madam. I can't understand Hindi.
You're from which place?
Karnataka, madam.
Okay, you're from Karnataka. Okay, sir. I just wanted to know whether you want us to conduct this kind of program or you are planning to conduct this program.
I telling you to conduct the programs, madam.
Okay. No worries, sir. We will surely take your feedback and we will surely conduct and we'll get back to you, sir. We have your email ID and we will surely get back to you.
When I hearing this webinar, you call the resource persons are very good, I think.
Okay.
And please conduct the webinars on taxonomy, physiology, pathology, these related aspects.
Okay, sir. Thank you, sir. We will surely do that. We will be surely taking up your feedback and going ahead with these programs, and we'll let you know. Yeah. Thank you so much, sir. Thank you.
Thanks,
Thank you
madam.
so. Thank you so much. Thank you. Ritu madam, are we done with the things?
Yes, madam.
Yeah, everybody has got the, I think, feedback, and I think there is no issue now, right?
Yes,
and I have taken the pictures also.
Yes.
Okay.
Should we leave the meeting?
Can you call Jayashree madam? I think Jayashree madam was not there for the photograph.
Okay.
Yeah. चारों हम लोग आ सकते हैं क्या? अगर हम लोग ये स्क्रीन का कुछ अगर चेंज कर देंगे तो हम लोग पूरा डिपार्टमेंट का.
Madam, is photo session over?
Yeah, the photo session is over, sir. We got your photo. We got your photo.
Thanks.
We got your photo. Yeah. Okay, we got your photo.
Thanks.
Okay.
Bye, madam.
Yeah. Thank you. Bye. Yeah. Okay. Ritu madam, can we leave now? I think जयश्री मैडम आ रहे हैं क्या? वैशाली मैडम,
जयश्री मैडम को मैंने कॉल किया है.
नाइट मैडम भी नहीं थे हमारे फोटोग्राफ.
नहीं, नाइट मैम ऑन वे है, तो मैडम बोले कि वो ट्रैफिक में अटके तो शायद वो ज्वाइन नहीं कर पाएंगे अपने को, वहां उनका बैटरी भी कम हो गया है तो वो ज्वाइन नहीं कर पाएंगे उन्होंने कहा है. बट मैंने स्क्रीनशॉट्स ले लिए हैं. वैशाली मैडम, आपने लिए हैं स्क्रीनशॉट्स वगैरह पहले के?
हाँ, मैं नहीं, पहले के नहीं ले पाई मैं.
लेक्चर के?
नहीं.
अच्छा, ओके.
जयश्री मैडम को मैसेज किया.
हाँ, रिकॉर्डिंग है ना, हमारा रिकॉर्डिंग है ना. या रिकॉर्डिंग है ना पूरा, नो प्रॉब्लम. मैडम रिकॉर्डिंग अभी आप स्टॉप कर सकते हैं. रीतू मैडम, रिकॉर्डिंग अभी
यस.
हाँ, स्टॉप कर दीजिएगा.